Swayam Prabha. Digital India. Educated India.
So, in today's class, I will continue with the subject of tolerance and immunity. I had already introduced to you what is autoimmunity. Just going back to the history, it was sometime in the early part of the 20th century that Dr. Paul Ehrlich realized that one can evoke an immune reaction against self antigens, and he termed this condition as autotoxicosis, because we always believed till then that the immune system has the capacity to mount an immune response only against non-self antigens. Now, obviously, if the antibodies or T cells start attacking self antigens, then naturally this would be a process which would be continuous, unlike in case of pathogens, where the immune system is activated only till the period that the pathogen exists in the body. There are several diseases that can be studied under this autoimmunity. Diseases like diabetes type 1, rheumatoid arthritis, systemic lupus erythematosus, SLE for short, Graves' disease. These result from the failure of the immune system to distinguish self from non-self. We will come to how this situation happens and how is it that a small percentage of people get afflicted with these conditions. Before that, we will just go over what are the different autoimmune diseases which are common. Now, autoimmune diseases can be both organ specific or systemic. Now, of course, as the name suggests, organ specific would be the immune response which targets unique antigens which are specific to a particular target cell or an organ, and this of course would cause cellular damage directly. There are 4 diseases that I am going to talk about under this. One is Hashimoto's thyroiditis, autoimmune anemia, Goodpasture syndrome, and IDDM or insulin dependent diabetes mellitus. Let us start with Hashimoto's thyroiditis. In this particular condition, of course, the organ involved is the thyroid, again as the name suggests. This is because of autoreactive sensitized Th1 type of cells to thyroid antigens. If you remember the class on hypersensitivity, you would remember that the type 4 hypersensitivity is because of T-DTH cells, which are Th1 cells, and these cells are the causative factor for Hashimoto's thyroiditis. There is intense infiltration of immune cells in the thyroid gland, and there is therefore an inflammatory response in the thyroid gland. Now, whenever there is T-DTH response, it is associated with inflammatory response. So, imagine that now all organs of course will have immune cells, for example, macrophages all the time. Now, imagine a case where there is intense infiltration of lymphocytes, which would be Th1 mostly, macrophages activated, as well as plasma cells, because in this particular condition there are also autoantibodies generated to thyroglobulin and thyroid peroxidase. Now, thyroid peroxidase is the enzyme which is required for iodine uptake, and therefore, if there are autoantibodies to the thyroid peroxidase, there would be inhibition of iodine uptake by the gland, and this would result the condition, the autoimmunity results or now causes hypothyroidism. Hypo would mean lesser amount of thyroxine secreted by the gland. Now, the second in this particular list of autoimmune diseases, which is organ specific, is autoimmune anemia. Autoimmune anemia again can be due to several factors. Let us start off with pernicious anemia, which is not very uncommon. Pernicious anemia is because of the antibodies to the intrinsic factor. Now, what is this intrinsic factor? It is a receptor which is present on the parietal gastric cells, and this allows the absorption of vitamin B12 from the diet. So, antibodies bind to the intrinsic factor, do not allow absorption of vitamin B12, and therefore, now vitamin B12 is required for hematopoiesis and generation of red blood cells. Now, therefore, in this particular conditions of pernicious anemia, there is lack of vitamin B12 and therefore, lack of hematopoiesis, adequate hematopoiesis, and therefore, resulting in anemia. So, the treatment for this type of autoimmune anemia would be supplementation with vitamin B12. So, it is important therefore, that the anemia is diagnosed and found that it is because of antibodies, and therefore, the only way would be supplementation with vitamin B12 in the diet. Hemolytic anemia, this can be caused because of antibodies that are made to modified red blood cell membrane proteins. If you might remember my lecture on hypersensitivity, some individuals exhibit this property. Their cells, RBCs, sometimes also could be skin RBCs, in this case of course, we are talking about red blood cells. The membrane proteins have the capacity to bind to certain small molecules, allowing now these small molecules which otherwise would not have been immunogenic to be immunogenic. Therefore, there would be antibodies to these modified membranes, and antibodies bound to the membrane now would attract complement cascade, trigger the complement to now make membrane attack complex, which lyses red blood cells, and the anemia is caused because of constant lysis of red blood cells. Now, because this is drug induced hemolytic anemia, the way to treat this would be to withdraw the drug. If there is no drug, there would be no drug which is retained on the red blood cell membrane, and therefore, there would be no now further cell lysis, because now red blood cells would not be modified. Goodpasture syndrome, this is another autoimmune disease of an organ particular cell. Antibodies in this condition, antibodies are found to the basement membrane antigens either on kidney glomeruli or lung alveoli. Again, in this particular case, though there are now, what does this condition? If there are antibodies, okay, if the antibodies are not harmful, does not matter. But what happens in these cases, the antibodies, constant generation of antibodies to these membrane antigens would now result in antigen antibody interaction on the membrane. This can effectively trigger the complement cascade. Just to recapitulate your memory with respect to this complement mediated activation, you might remember that complement activation leads to now activation of the complement components, the starting with C1q, then you have C4, C2, C3, C5. You might remember that until the component C5, there is cleavage of an inactive form of the component to an active form, and the release of small components, as happens with C4, C3 and C5, that the cleavage of the pro enzyme to an active enzyme and the release of these small factors C3a, C4 and C5a, this causes release of granules from mast cells, because these bind to specific receptors on the mast cells and basophils. So, therefore, this would now, these are known as also, you might remember, anaphylatoxins, because they are able to induce a hypersensitive type reaction, though slight milder, but they do evoke that response. Apart from that, apart from the inflammation that is caused because of these molecules, not only do they are they anaphylatoxins with respect to release of pre-filled granules of in mast cells, but they also are chemotactic factors. So, therefore, wherever the complement where antigen antibody interaction takes place, in this particular case it would be the membrane antigen of kidney glomeruli and lung alveoli. At that site there is attraction of several immune cells, neutrophils, macrophages, etc. Finally, there is also cellular damage caused by the formation of membrane attack complex. Now, the membrane attack complex is not very specific with respect to what it kills. All bystander cells would get killed wherever the MAC gets deposited, a membrane attack complex. And in this case, because the antigen antibody interaction is taking place on the membrane cells, these cells get attacked and these cells lyse because of MAC. The disease can be fatal because if not treated, progression of the disease leads to kidney damage, because we are talking about the glomeruli, as well as if in the case of lung alveoli, then you have pulmonary hemorrhage, both the conditions being fatal. Let us come to now insulin dependent diabetes mellitus. Insulin dependent diabetes mellitus is also known as juvenile diabetes, and if it affects about 0.2% of people, and it is called juvenile diabetes because we know that it usually affects children. This happens because of immune attack destroying beta cells of islets of Langerhans. So, islets of Langerhans are the cells which synthesize insulin. Because the beta cells themselves get destroyed, the amount of insulin that is produced is much lesser, and this leads to diabetes. This again can be mild versus severe. Of course, in severe cases, there is almost no insulin production. What is this autoimmune disease because of? Apart from, because of. Now, what are the kind of cells that are seen to migrate to the pancreas? It starts off with large number of cytotoxic T cells, or CTLs for short. Because of, there is also local cytokine production, interferon gamma, TNF alpha, interleukin 1 which now activates macrophages. There are also at a later stage of this disease autoantibodies to certain antigens on beta cells. So, therefore, now the inappropriate deposition of large number of immune cells to that site and activated cytotoxic T cells and antibodies to antigens of the beta cells now causes damage of beta cells, lowering number of beta cells and thereby lowering insulin production. These individuals have to be supplementation with insulin. Now, we have talked about immune system which is directed to particular organ or a particular cell. We now come to autoimmune diseases which are caused by antibodies that may act as agonists or antagonists. Let us look at what this means. Agonists, which would mean that in case it is a hormone or a lichen, these antibodies act synergistically to that hormone and increase whatever is the ends or the result of this hormone action. In this particular situation, in this case of an antibody being an agonist or has an activity which is the same as that of the hormone, the example is Graves' disease. in this particular condition, there are stimulating autoantibodies to antigens on the thyroid cells. Let us look at what happens in this condition. There are antibodies to the thyroid stimulating hormone receptor. The TSH receptor is present on thyroid cells. Okay. Now, the TSH receptor receives thyroid stimulating hormone, which is synthesized and secreted by the pituitary. TSH binds to TSH receptor, and this triggers a signaling process, which now leads to production of thyroid hormones. The thyroid hormones themselves can inhibit further production of TSH. So, there is an extremely, many of these endocrine systems have a negative feedback. So, there is a loop. So, pituitary synthesizes TSH, TSH binds to its cognate receptors present on thyroid cells, inducing signaling, which now results in the release of thyroid hormones by the thyroid cell, increase in this particular circulatory level of the hormones, inhibits for the production of TSH. So, it is an extremely tightly regulated setup, a network between pituitary and thyroid cells. In Graves' disease patients, there are antibodies to the TSH receptor. So, what do these antibodies do? Now, I must tell you that not all antibodies are able to trigger. You might remember when we studied signaling in B cells, you might remember that the way signaling in B cells has been studied is by antibodies to the receptor, antigen receptor on B cells. If you have an antibody to an immunoglobulin molecule, this would bring about cross-linking of receptors, two receptors with one antibody on the B cell, triggering a signaling process which is very similar to that seen when an antigen binds to the B cell receptor. The same situation happens here. These individuals develop antibodies that can cross-link receptors. Now, these antibodies cross-link TSH receptor, inducing a signaling which is very similar to what happens when TSH hormone itself binds to the receptor. This would result therefore in production of thyroid hormones. Now, the production of thyroid hormone is now independent of the TSH binding to its receptor. Therefore, even though thyroid hormones, as happens in the normal case, would bind to the pituitary cells, the cells in the pituitary which release TSH, the TSH may not anymore be made, but in this particular situation, production of thyroid hormones is totally unregulated, because the feedback loop, even though it is there and there is no further production of TSH, presence of antibodies themselves would keep on triggering in an unregulated fashion the activation, the signaling and release of thyroid hormone. So, Graves' disease patients show symptoms of hyperthyroidism. So, we have just finished dealing with Hashimoto's thyroiditis, where there are antibodies to the peroxidase, which now inhibits thyroid hormone production by the thyroid gland. Now, in this same situation, in a situation where thyroid gland is involved, but here autoantibodies to the receptor now induce a condition which is opposite of Hashimoto's thyroiditis. Now, these patients show symptoms of hyperthyroidism. Just to go a little bit in details with respect to the antibodies. Now, the TSH receptor belongs to the 7 transmembrane receptor family. When there are 2 such receptors, as shown in the same picture here, associated with one with dotted lines, that is receptor number 2. There are antibodies in the same patient seen to the extracellular loops of the TSH receptor. Now, these loops are the ones which extend in the cell in the aqueous phase, they are present on, they extend into the outside of the cell, where there is free access to these antibodies. And you can see here 2 receptors, the loop 1 of both 2 receptors are being cross linked by antibody. Now, the red antibody is different from the green antibody, which is to the second extracellular loop, and the third, which is to the third extracellular loop. Now, it has been seen that there could be variation in the kind of triggering that happens, but all 3 antibodies to all 3 loops have the capacity to trigger signaling. Let us come now to antibodies that are antagonists. We have talked about the antibodies to TSH receptor, which act as agonists. We talk now about blocking antibodies, which inhibit a signaling process. The example that I would like to introduce here, talk about is myasthenia gravis, a condition where there is muscle inactivation. Now, what I have shown in the picture here is a nerve cell, which is in close proximity to the muscle plate, and this is the neuromuscle joint. When there is activation or an impulse of the nerve, there is secretion of acetylcholine. The acetylcholine molecules bind to their cognate receptors. These are on the terminal ends of the muscle plate, there are these receptors to acetylcholine. This binding of acetylcholine to its receptor is what brings about muscle activation. In case of myasthenia gravis patients, they develop antibodies to the acetylcholine receptors, which block the binding of the acetylcholine to the receptor. This results in loss of activation because there is no ligand receptor interaction. Since signaling is inhibited by antibodies, there will be no neuromuscular, there is no muscle activation. Also, with large number of molecules of antibodies bound to the acetylcholine receptor, there is complement activation. As you know, IgM as well as IgG isotypes can activate the complement, and we just discussed a couple of minutes ago that this activates the complement, which can result in destruction of the acetylcholine receptors themselves by the membrane attack complex. So, receptors get destroyed, and as this disease, if not treated or even treated, eventually leads to paralysis. Now, we will come to systemic diseases, autoimmunity, which affects the entire system. The first one under this is a not so common, but known condition called SLE, systemic lupus erythematosus. This is characterized by fever, well mild, but constant fever, skin rashes, arthritis, kidney dysfunction. The name, the name lupus, actually individuals with this condition are characterized by a butterfly kind of red patch on the face. These individuals have antibodies to DNA, their own DNA of course, histones, red blood cells, platelets, leukocytes, clotting factors, and the condition leads to anemia, thrombocytopenia. Anemia because red blood cells get destroyed by the same path that I have been mentioning, that you have antigen and antibody interaction and activation of the complement, which would lyse the target cells. Thrombocytopenia, when the antibodies are to blood platelets, there is depletion of platelets in the circulation. I have already said that is because immune complexes which are deposited along blood vessels activating the complement. Not only is MAC or membrane attack complex formed, which destroys cells, but there is also hypersensitivity type 3 reaction because of immune complexes that are deposited all along blood vessels, and that means that is inflammatory. Now, we come to a disease which is more common than the ones that I have talked about so far, rheumatoid arthritis. This is a disorder where individuals develop autoantibodies of IgM class to IgG. I have already introduced briefly to this particular condition when I talked about the structure of immunoglobulins. Now, how is the structure of immunoglobulin associated with this? I will come to that, but the part on the FC region of of the IgG is on the FC region. Now, that particular domain of IgG, mainly CH2, which is glycosylated. This might ring a bell. I talked about the fact that glycans and differences in glycans in certain individuals can bring about the establishment of or availability of an epitope on the FC region, which otherwise would be hidden or covered by the glycans. So, you have now glycans which are present on the CH domain. In case of such individuals who have rheumatoid arthritis, in these individuals there is lack of a lack of certain enzymes that add sugars and make this particular chain of glycans. So, because of that, the region on the FC part of IgG gets exposed to immune cells, and there are autoantibodies usually only of IgM type, which now complex with the IgG in circulation. So, there are small complexes that are formed all the time. These would get deposited well in blood vessels, but also in joints. Once again, as in previous cases, these immune complexes activate complement, resulting in hypersensitivity type 3 reaction. Now, I will just introduce this particular conditions, because there will be another the lecture on T cell immune deficiencies will actually deal with this in more details. But just to mention, multiple sclerosis is a condition where there are autoreactive T cells to the myelin basic protein. MBP is this MBP is present in the myelin sheath. As you all might know, that myelin sheath is what insulates the nerves. When there are normally T cells like B cells do not cross the blood brain barrier, but activated T cells can. So, when there are autoreactive T cells, they can enter the blood brain barrier and they injure the myelin sheath. This causes neurological dysfunction, because once again the insulation is lost. Like I said, this will be dealt with in details, this disease. So, in summary, with regard to autoimmunity, not all self reactive, I would just like to reiterate that though in the start of my course, especially when I talked about the development of B cells, I did mention that in the bone marrow, B cells that are developing to become from pro B cell to mature B cell, there is a stage where they have only IgM receptor, antigen receptor present on the cells. Now, before they can acquire IgD type of receptors on the cell surface, these cells can undergo apoptosis if either the receptors are cross-linked, as is done experimentally, but in the situation in the bone marrow, if they come across and bind to self antigens. Now, at that time one can imagine that there would be only self antigens which are circulating from the bone marrow to the periphery. In spite of this check, where autoreactive B cells should be deleted, it is known of course that there are a small number of cells, B cells, that carry receptors that have the capacity to react with self antigens. These are released in circulation, but are kept in an anergic state. However, in certain conditions, and we will see what those conditions are, these very cells which are quiescent get activated and cause autoimmune disease. Are there any animal models to study autoimmune disease? Because of course, autoimmunity can be a condition which can be of course fatal. There is also this condition can be very chronic. To be able to understand how this autoimmunity develops, one needs to have animal models, and there are some experimentally induced conditions versus spontaneous. Let us first talk about the spontaneous one. Now, there are strains of mice, the first one being non-obese diabetic mouse. This mouse shows or develops insulin dependent diabetes mellitus. This develops spontaneously, and this is a very good animal model to understand IDDM. The second is an F1 mouse, a cross between New Zealand B mice and New Zealand W mice. These mice develop spontaneously the condition of systemic lupus erythematosus. So, these mice have autoantibodies to RBCs, platelets, DNA, histones, and conditions similar to what one sees in human. Many mice models are available where one can induce experimentally conditions of myasthenia gravis. This can be in mice by injecting acetylcholine receptor, so that antibodies to acetylcholine receptors are developed. Now, experimental EAMG is experimental autoimmune myasthenia gravis. When mice are injected with MBP or myelin basic protein, they develop experimental autoimmune encephalitis, which is similar to the human multiple sclerosis. Similarly, injecting mice with Mycobacterium tuberculosis bacteria induces in these mice symptoms similar to rheumatoid arthritis. They develop autoimmune arthritis. Also, there are mice develop Hashimoto's thyroiditis. So, there are these animal models which have been established, and they have helped understand autoimmune diseases. So, there have to be mechanisms which induce autoimmunity, because we know that fortunately very few of us are afflicted with these conditions. The proposed mechanisms for induction of autoimmunity are release of sequestered antigen, molecular mimicry, which explains most of the autoimmune diseases, inappropriate expression of MHC molecules, polyclonal B cell activation. Let us start with release of sequestered antigen. The examples are myelin basic protein, sperm antigens, and lens of the eyes. Now, we go back again to self versus non-self. Now, in this particular situation of like MBP, there is a blood brain barrier because of which MBP is totally sequestered in the on the side of the neurological system, and therefore, MBP is never allowed to enter the bone marrow where negative selection takes place. Now, this is fine as long as there is no exposure of MBP to the immune system. This happens in case of injury to the spinal cord, which may release MBP into circulation, and thereby now T cells would get activated and subsequently also B cells. And now activated T cells, as I told you a little while ago, activated T cells can cross the blood brain barrier and now start to cause damage. So, MBP under normal circumstances would be a sequestered antigen, but this is broken because of injury. Sperm antigens. Again, there is a testis blood barrier. Therefore, sperms, which in fact are the cells which are made only after puberty. Sperm antigens are again sequestered in the testis, but either injury to the testes or in case of vasectomy, there is sperm entry into circulation, and these are of course foreign to the circulatory system, and therefore, antibodies are made. Some individuals who have been vasectomized, but would like to reverse the process, have been proven to be infertile because they develop antibodies to sperms. Again, in the case of lens of the eyes, there are different proteins of the lens. Usually again, there is a barrier between the anterior chamber of the eye and the circulatory system. Injury to the eye causes exposure of lens to immune cells, and there are autoantibodies which can actually attack the crystallines. So, sequestered antigen is one which is very, of course, very easy to understand, and this has been shown experimentally that if one now in the time of development, if let us say an animal is a piece of the lens is now placed in the thymus, it is seen that such animals too would not develop autoimmune disease, all right, to the crystallines or the proteins of the lens. So, therefore, sequestered antigen, that is it is understood very well that anything that breaks the barrier which keeps the sequestered antigen from being exposed to the circulatory system may lead or would lead to activation of the immune cells to those sequestered antigens. Second mechanism is molecular mimicry. There is much more data, and that what I have put on the slide here, but molecular mimicry is what explains most of the autoimmune conditions, where there are antigenic epitopes that our antigens share with bacterial proteins or bacterial or well not just bacterial, also viral components. Example of molecular mimicry is there are several examples. Streptococcal antigens cross react with heart muscle. Now, in certain conditions of heart failure, it has been shown that those individuals, well, it was realized that those individuals had had streptococcal infection prior to the heart attack, just recent past, in the recent past. Now, people started to look at how streptococcal infections, or whether there was any relationship between streptococcal antigens and the heart muscle, and found that there are certain epitopes which seem to be conserved in the two. So, therefore, a prior exposure to the streptococcal antigens would evoke an immune response, and because there are antibodies to these streptococcal antigens, they can attack the heart muscle, which also carry the same epitope. Just to give you how closely related these epitopes may be, now here I have 5 different pairs. Human cytomegalovirus 1E2, a sequence of this particular protein in the cytomegalovirus shows almost 100% identity with respect to 5 of the amino acids with HLA-DR molecule. So, again, if there are antibodies, which if one generates antibodies to this particular region, then those very same antibodies can now bind HLA-DR. Poliovirus VP2, you can see the similarity with 6 amino acids of the VP2 of poliovirus with the acetylcholine receptor. So, now this becomes quite easy to understand. If one has been affected, one has been infected with poliovirus VP2 even without realizing, if one has made antibodies to this particular region, then that can bind to the acetylcholine receptors and could serve as blocking antibodies. Same is the situation with papillomavirus E2, which has antigenic determinants similar to insulin receptor, HIV protein P24, you can see 100% identity with the human IgG constant domain, measles virus P3 with MVP. So, you can see with the number of, now this also suggests that fortunately again these may not be immunogenic, or not all individuals would make antibodies when infected with human cytomegalovirus or poliovirus or papillomavirus. These may not, are certainly not the immunodominant epitopes, either B or T cell epitopes. These are minor epitopes, and only certain individuals by chance would make antibodies to these. Therefore, it is only in certain individuals that if one has made antibodies to certain regions or antigenic epitopes in the infecting organism, to the infecting organism, then there is a chance that these antibodies would now cause autoimmune diseases. The third one is inappropriate expression of class 2 molecules. This explains the destruction of beta cells. Class 2 molecules, we have been discussing all along, class 2 molecules are expressed only on antigen presenting cells, which are B cells, which are dendritic cells and macrophages. Now, class 2 molecules are not expressed on any other cells, but can be expressed if there is local concentration of gamma interferon. Now, presence of gamma interferon can activate the expression of class 2 molecules in cells which normally would not have. Beta cells from healthy individuals express only MHC class 1 molecules, because all nucleated cells express class 1 molecules. But it was observed that from IDDM individuals, insulin dependent diabetes mellitus individuals, class 2 molecules also express on beta cells. Now, this gave an impetus to start to look at what would be the relevance. So, similarly now thyroid acinar cells from Graves disease individuals express class 2 molecules. Mitogens such as PHA, so it is not only interferon gamma, experimentally it has been seen that even mitogens such as phytohemagglutinin, which activate T cells, can induce expression of MHC molecules on non-antigen presenting cells like epithelial cells. Now, PHA of course is a plant product. This is experimental, but it has been shown that gamma interferon can induce expression because the MHC molecule in the promoter region has a gamma interferon binding site. So, think in terms of a viral infection, and therefore, the earlier slide which showed, let us say infection with papilloma virus, which induces antibodies, let us say, to this particular region, then the papilloma virus infection itself can bring about interferon gamma production, which can result in inappropriate cells expressing class 2 molecules. Now, class 2 molecule expression would make those cells non-APC cells APCs or antigen presenters. Therefore, now they would present antigen to T cells which are self, and therefore, this activation of autoimmunity is triggered. Trauma or viral infection causes localized gamma interferon secretion, leading to class 2 expression on cells. This would now, especially in case of IDDM patients, it has been seen that their beta cells now start to express class 2 molecules and become antigen presenting cells, and these now induce the immune response to actually delete them from the pancreas. Inappropriate expression of class 2 molecules has been studied experimentally by making transgenic mice. What this experiment shows, that the insulin structural gene, from the insulin structural gene, the insulin gene promoter is amplified. So, also of course, the insulin gene terminator. Now, this is now put in frame with the interferon gamma gene. The insulin promoter now if mice are made transgenic with this particular construct, then because of the presence of insulin promoter, the gamma interferon gene would, this entire gene would be now targeted to the pancreas because of this insulin gene promoter, and this is where gamma interferon is produced. Such transgenic mice have been shown to develop diabetes by the mechanism that I just discussed, that gamma interferon is produced localized in the pancreas, in the beta cells, because of the presence of insulin gene promoter, and the gamma interferon now makes the pancreatic cells express class 2 molecules. These become antigen presenting cells, and therefore, there is an immune response evoked to the pancreatic cells, destroying the pancreatic cells in these transgenic mice and resulting in development of diabetes. So, this is a fine experiment that was carried out to show unequivocally that gamma interferon production induces class 2 expression, and of course, people have, in this paper which describes this experiment, people have gone on to show the abnormal expression of class 2 molecules on cells which are not antigen presenting cells. So, this would support the autoimmunity theory of inappropriate expression, which is induced by viruses. Viral infection does lead to expression or accumulation of gamma interferon, which in that local compartment would make non-APC cells express class 2 molecules and therefore becoming antigen presenting cells. What are the treatments of autoimmune diseases? Obviously, the best treatment would be aimed at reducing only autoimmune responses, leaving the immune system intact. But that is not successful, that is not even possible. So, the current therapies are only palliative, that means they only reduce the symptoms. This is done by using general immunosuppressive drugs such as corticosteroids, which affect the interleukin 2 signaling, as a thioprine cyclophosphamide, which inhibit proliferation of cells. So, this would be at the level of signaling and this would be at the level of proliferation. Cyclosporin is one drug which is used on antigen activated T cells, because cyclosporin again it inhibits in a very specific manner the triggering signaling of T cells. Finally, plasmapheresis can also be carried out as treatment, but all these are just palliative, they just decrease the symptom. Plasmapheresis, the plasma is put through a machine where immune complexes are depleted and therefore, there is elevation of symptoms, but patients need to undergo plasmapheresis from time to time. During the course of my lecture, I did not mention gender bias with respect to autoimmunity, but it is known that women are predisposed to developing autoimmune diseases than men. In fact, if the ratio of male to female or female to male is observed in rheumatoid arthritis, multiple sclerosis and Hashimoto's thyroiditis, it is seen that the ratio is 3:1, 3 female to 1 male, and in a system like SLE, it is 9:1, that ratio is extremely high. So, women are more prone to autoimmune diseases than men. So, there are possible explanations and also experimental evidences, experiments in mice which show that females produce higher titers of antibodies than males. So, that means they are immunologically more proficient. Women have higher levels of CD4 T cells and also have higher levels of serum IgG than men. Now, these are of course, these are direct evidences, but gender bias can be due to sex, obviously therefore, gender bias is because of sex steroids. So, what does estrogen do? Estrogen stimulates autoantibody production in SLE mice, SLE prone mice, and interestingly this can be reversed with anti-estrogens. So, here is an experimental evidence that estrogen, the female hormone, is directly correlated to autoantibody production and therefore, there is a gender bias. More interestingly, female non-obese diabetic mice are more susceptible to spontaneous diabetes. We already went through the non-obese diabetes mice which develop autoimmune disease by developing IgDM, but female NOD mice are more susceptible. Also, castration of these female mice castration of male mice, which would mean that testosterone is acts opposite to estrogen. So, castration of male mice, male NOD mice significantly increases the susceptibility in these. So, again direct correlation of estrogen and autoimmunity. Also, SJL mice which are susceptible to developing EAE or experimental autoimmune encephalitis, female mice are much more susceptible than male. Steroid hormones play a role in gender bias. Steroid receptors transcription factors. Immune cells have steroid receptors. So, this is the way one can explain the gender bias. Also, now prolactin, a peptide hormone which is of course a hormone of both males and females, but this is regulated by estrogen and therefore expressed in higher levels in women than men. Again, T and B cells have prolactin receptors. So, not only steroid receptors, but also prolactin receptors. Prolactin turns cells towards Th1 dominated immune response. So, once again you can see that there would be a gender bias. Just like to inform you that during pregnancy, the Th1 predominance is altered to a Th2 type. So, we now come to another aspect of immunological disorders. Now, we have talked about autoimmunity, the reasons for autoimmunity. Now, let us come to another immunological disorders, and I will restrict myself to B cells, are immunodeficiencies. Immunodeficiencies of B cells affect both the lineages of the lymphocytes. This causes severe combined immunodeficiencies. SCID can be caused by different genetic factors. In SCID, all forms of SCID have low numbers of lymphocytes. Even the low number of lymphocytes, even if they are present, they do not respond to mitogens. Mitogens are those proteins or molecules that can induce proliferation of T and or B cells, and you know that proliferation of T or B cells is a prerequisite activity before the cells differentiate to their effector population. So, in SCID, even if there are few number of lymphocytes, the few numbers themselves are not responders, and because of lack of either number of cells or inadequacy of these cells, there is severe recurrent infections, which usually is fatal and in early years of life. Defects that underlie SCID, the most common one is mutation of the interleukin 2 receptors, the gamma chain. This affects signaling of receptors also for interleukin 4, 7, 9 and 15, because this is the common chain between all the receptors. The other defects that underlie SCID is defects in RAG1 and 2. Now, in case of defects in RAG1 and 2, you know immediately that this would not allow generation of T or B cells. In certain individuals, there are normal lymphocytes, but signaling is affected totally. These do not have or have deficiency in a kinase called ZAP70, and therefore, there is inefficient signaling. Now, there are two others which I will go over quickly: X-linked A-gamma globulinemia, Bruton's hypoglobulinemia, because there is defect in BTK, Bruton's tyrosine kinase. In these individuals, the B cells remain in the pre-B cell stage and there is no rearrangement of the heavy chain. In case of X-linked hyper IgM syndrome, these individuals have elevated levels of IgM and lack totally IgG, Ig and IgE, because these individuals lack CD40 ligand expression. So, I will like to stop here and I will continue in my next lecture with a part of these immunodeficiencies and then go on to cancers, which can also be called immunodeficient disease. Thank you.